what I'm going to do, actually I'm not only going to be talking about the subjects that I sent months ago on the third lecture, because I was filled with enthusiasm for the 3 topics and I realized I didn't know enough to give 3 lectures. I should ask her, do we break or do I just go for 90 minutes?
We can break if you want, and there's a tea break at 10:30.
Okay, so I think it would be appropriate if I told you a little bit about my background. I was trained as a biochemist, so this is actually the right building for me to be in. And as you know, biochemists put great stock by experimental data. They like to track down every last molecule and make sure they understand it before they do anything else. So that was my background. And then about 20 years ago, I realized that there was some limits to what I was doing. I was doing mainly genetics. And I kept running into things, one of which I'll talk about in the third lecture, things that really required some quantitative thinking, quantitative reasoning. And I think that for me, that's what physics and physicists bring to biology. It's not so much the tools they have, although they're important. I mean, I'm an expert on calculus. I mean, I failed introductory calculus twice as an undergraduate, so I feel I know a lot about it. But those tools are very important, but the qualitative skills that you develop as a physicist to think things through carefully and not give up till you really understand them. Biologists are not very good at that. Once they have described something, they tend to move on because they realize how complex it is. They don't hang on to it like a dog with a bone until they really feel they've mastered it. So when I began to do this, it also became clear that the students that I would have to have working with me would probably be applied math and physics students. And the first one was an applied math student at Princeton. And then since then, the last 10 years, they've been mainly PhDs or postdocs in physics. And some of the most successful ones, and I'll be talking about his work today, came from as a condensed matter theorist. And I found that that worked really well because I told him how to do experiments and he didn't dispute my word because he didn't know anything about laboratory science. Whereas the applied physicists that I had in my lab, I've had quite a few, they already thought they knew how to do experiments. And so sometimes it worked, sometimes it didn't work. So I think that, I don't know how many of you are theoretically inclined people, but one of my things that I've been saying over the last 10 years is that it's really important, if possible, to get some hands-on experience to learn the gap between theory and practice. So I'm going to pick 3 topics to talk about while I'm here. And one of them is, and first I want to thank the organizers for inviting me. This is a very distinguished part of science around the world. I've heard about this institute long before I visited, so it's had a big influence on science in general. I'm going to talk about mRNA synthesis, one molecule at a time. And the take-home lesson is, and I hope not too many of you know this, or I'll be boring you, is that contrary to the models that existed about 10 years ago, messenger RNA synthesis, the copy you make from the DNA, is in fact more complex than we thought. Certainly more complicated than I thought as a biochemist. Because a biochemist breaks everything apart and studies it in a test tube. And by then, you've lost the living cell. So the 3 things I'm going to be emphasizing are done with live cells. 2 of them, that's 1 and 2 here, mRNA synthesis, one molecule at a time, and then finding a target in a crowded cytoplasm. I'll be talking about how a repressor molecule finds its target on the DNA, and I'll also give you some experiments that have a bearing on how crowded the cytoplasm is. And another thread that goes through most of this actually is in fact diffusion. I had a talk prepared for my own faculty called Diffusion and Me, because at one time in my lab about 5 years ago, someone was doing super diffusion, someone was doing subdiffusion, and I was doing true diffusion. Not just my ideas, but in the laboratory. And I'm also going to give you some basic introduction to the material because I went around our physics department talking to graduate students and postdocs who do biology, asking them about their background, and it turned out it was pretty shallow, and they had some confusions. So you'll have to bear with me because some of you know this, but some of you probably don't. And my main goal in all of this is to persuade you to realize how difficult it is to do experiments and how tentative the answers are. And that's partly so you don't go completely hog wild doing theory with a very ill-constrained data set. So my heroes in physical biology, I won't mention them, but who come from physics, seem to have this uncanny ability to know when the data set is constrained enough that they can fruitfully go in there and make their contribution and deepen our understanding. I think that's a real trick and not so easy to do. So let me get started with topic 1, which, well, that's some reading I sent ahead. And I'm going to, by way of talking about E. coli, I'm going to show you where this is all headed with dictyostelium, which won't be till the 3rd lecture. But I'm going to show you some movies so that you'll come back for the 3rd lecture, basically. So I don't know how many of you understand this, so I'm just going to be very basic. Dictyostelium is a free-living soil amoeba, and it feeds on bacteria and yeast. You can go out around this area here and find all kinds of different species. And they have a remarkable self-organizing property, and that's what some of this is going to show you. When they run out of food, here's one actually hunting for E. coli. And one of the topics I'll be talking about is what kind of motion, the ground state for amoeboid motion, before you do anything, before you sense a prey or before you begin to cooperate with your neighbors, what do you actually do? And it turns out to be very interesting. It's a persistent random walk of a certain kind in which the cells zigzag. They turn left and right, but with time they drift. And so over a longer time scale, things are truly diffusive, but in a short time scale, they show this left-right choice. So here's a single amoeba. It's actually, you can see it hunted down. This is taken by Simon Fligeberg, whom Norelka sometimes calls himself. You may know him. His dad's a famous guy. So here's a tiny colony of E. coli. So this is only, so this amoeba is about 10 microns in diameter. So this is a very tiny colony of E. coli. And this, no one has pursued this because it has some remarkable properties. And you'll see that at the end. So they come up, they begin to eat, and when they're full, they divide. You saw that happen. One came up. It's just a fluke that we got. But notice the others have run away now. They've decided that there's no longer any food there. And then this last one was a bit slow to learn that lesson and left kind of, so there's a kind of repulsive force that sets in at some point too that we don't understand. So it's complicated. But there it goes. 2 of them now, they become 4. Here's some neighbors come in. Okay, so that's enough for that. And I want to show you now some of the self-organizing properties. And this was done by Rob Cooper, who's now at UCSD doing his postdoctoral studies. This is just a petri dish covered with E. coli. And this is essentially a wave of amoeba and bacteria. So this is solid bacterial food. The dictyostelium cells are over here. And I want you to actually look at a couple of features, some of which are well understood, and I think we made a contribution there, and a lot that aren't. So you'll notice there's this teeming mass of individual cells. You're looking at maybe 10 to the 5th to 10 to the 6th on the left side of the field. And with time, they leave masses behind, and these masses begin to migrate around. And then finally, they differentiate into only 2 cell types, spores at the top here, and stalk cells here. Here's one. They're looking for kind of a dry space. Somewhere around here, you can actually see waves. These waves are cyclic AMP waves. They're being secreted in this coordinated fashion. And I'll spend most of the 3rd lecture on what we know about that and how we understand it. So these guys have dead stalk cells, and they've pushed up a ball of spores here. And those spores can now blow over and get started again on fresh bacteria. So that's just phenomenology, but it's important because these are the things that we seek to understand. There's not very much known at all about how the slug moves and why it moves the way it does, and there's very little really understood about the proportioning between these dead stalk cells and these spore cells. We know that roughly a third of the cells become dead stalk cells, and the other 2/3 become spores up here. But when we actually ask how this, let's see, this guy, how it makes that proportioning, and especially we know something about the mean, but we know nothing. The variance hasn't really been measured, and it hasn't been modeled in any sense that you would understand as a model, although people have claimed to do some modeling. Okay, so I hope that'll bring you back for the third lecture, and I'm going to talk about E. coli. And I'm going to talk about E. coli because it is far and away the best understood single-cell organism around. You can do genetics with it in a way and with a speed you can't do, even in yeast. And it allows you to pose questions and answer them, I think, in really precise ways, and then to model them because you can get huge amounts of data. So this is, again, a 10-centimeter plate of E. coli cells. These are colonies. If you've worked, how many actually you've worked with coli in the lab? And show of hands. So you probably know the difference between the blue and the white ones. One of them can ferment lactose and the other one can't. And I'm going to be talking about how these cells make message in response to this inducer. This is just a lactose molecule here. And this molecule is cleaved hydrolytically, and that allows the colonies to grow if lactose is the only carbon source. Okay, so a brief review for those of you who haven't thought about this or know very little about it, because I'm going to be talking in detail now about how the cell chooses to make this step, which is to bind RNA polymerase to a promoter site on the DNA and then get started to make messenger RNA. It's really a two-part process. The protein has to find the site, and then it has to be activated or inactivated depending on whether the system of control is positive or negative. This just shows in more detail something I'll be talking about in quite a lot more detail in Lecture 2. That is that the promoter region, so-called because it promotes messenger RNA synthesis, binds the RNA polymerase here, and here's a repressor molecule that can stop this from happening. This happens to be taken from what I call the baby version of Alberts et al., the big cell biology book, and it has to do with tryptophan synthetase, which in fact activates the repressor by binding tryptophan, whereas the example I gave you, you inactivate it by binding a product of lactose fermentation. And this is just another larger view of it. So the things that we are trying to understand then is how we pick this site, what are the energetics of it, what are the dynamics of it, and I'll get back to that a bit later. For those of you who want to look at eukaryotic cells, such as yeast and mammalian cells, the situation is much more complex, and it's the reason I stick with E. coli for these kinds of questions. In fact, Roger Kornberg, who was my very first student, got a Nobel Prize for saying that indeed this mediator is really there, but it's about 20 or 30 proteins. So it's a complicated business. There's an awful lot of biochemistry that isn't yet known about this structure, although we do know a lot about these enhancer sequences and various regulators. But it's a good reason to stick with E. coli, I think, until we understand it. Okay, back to E. coli. This is a famous picture taken by Oscar Miller, a long time ago now, and it shows that actually the messenger RNA is being translated by ribosomes as it's being made. So this is the direction of transcription. The polymerase got on somewhere up here, and with time, this messenger RNA grows, and as soon as it's available, a ribosome gets on, and then the ribosome makes some protein. This all takes place in a very compact, crowded cell. And so I want to talk for the rest of today about how these choices are made. And I'm going to show you some methodology because it's at the heart of what I'm going to say. And it is how we detect single message molecules. Now, a lot of biochemistry has been done on this system over the years with extracts of E. coli and a lot of other organisms, but we decided to see if we could actually catch it in living cells. And this is not an easy thing to do, and I'll come back to that too later. It's hard to see single molecules of anything in a living cell. And I think it's fair to say that the techniques are still being developed for this. So here's the way we did it. We took advantage of a technique originally designed by Rob Singer and published in '98, where you put a synthetic structure at the end of the gene you're studying. So in our case, we're going to take the lac gene for lactose fermentation. There's a promoter at this end. The polymerase gets on here. This is the 5' end. We begin to make RNA. Transcription goes in this direction. And then at this end, we've put a series of hairpin structures. In our case, we use 96 because we want to amplify a signal. And the way we do that is we take a second construct, which is a coat protein from a bacteriophage called MS2 that's being fused to green fluorescent protein. And in the background of the cell, we make a low constant level of these guys. So the cells are pale green. I like movies, so you'll see more of this. They're pale green. And then as soon as this is completed, these guys can bind, and you get a bright spot. They never bind 96. They usually bind about 50. And that becomes important when you do a quantitative interpretation of our data. So this is the system. And now we can see this quite easily, actually, with an ordinary fluorescent microscope. All right, so I'm going to stop and take questions because I got a feeling that I have a bad habit. I've spent most of my life talking to 300 pre-meds at once in a big lecture hall, so there's no time to stop. But I don't have to do that here. So, questions? Clear so far? Yeah. These guys?
Yes. So when you have the polymerase going across, they'll unbind, or what? Is that the...
No. It first completes the message.
Okay.
And releases it.
Ah, okay. Sorry.
Then,
sorry. I thought it was the DNA structure you were manipulating.
No.
It's the mRNA structure. Okay.
The DNA structure that just codes for these 96.
Okay, I got it.
And as far as we know, it can't bind because it's double-stranded DNA. It doesn't get a chance to loop. Maybe it does when it's being replicated, but basically it's just the way I've drawn it. So we make it, and then we bind it. And you can do this a couple of ways, right? Obviously, here you're looking at the last step, because it's at this end. But all the experiments we've done and others have done is to put these up here as well to see what happens first. Yeah, in the back. I'm going to need a microphone there.
How is the stability of the mRNA being maintained? Because they usually have these poly A tails at the end as well.
Yes, okay, so that's important actually for the stuff that'll come later, but basically it immortalizes the RNA. So they get just passed on in the daughter cells, and they don't turn over, or they turn over so slowly that we don't see it.
And the second question is, how do you maintain the background distribution of GFP uniformly throughout?
Yes, so that's pretty straightforward. But it's an important one. So here's an E. coli cell. And there's my structure here. And there's our hairpins. Okay? And over here we have a plasmid that codes for the GFP MS2 fusion. So it's always being made constitutively at a low level. And I've got some nice pictures that show that. So the cells are this pale green because it's everywhere. And then it gets trapped on these hairpins to produce the bright spot. Okay? Good. Those are good questions. Okay. So here's a movie. It was taken just a couple of years ago by Ido Golding, who did most of these experiments in my lab. So on the left, we're just watching the cells divide, and here's the time. So there was about 3 hours of time, and you can follow lineages here. You see, don't be misled by these patches. It's just where the cells are pressed up against the cover slip underneath the microscope objective. And then if you were watching carefully here, you can see bright spots coming on. But first, you just want to notice that there are bright spots, and that's when these MS2 GFP molecules become attached to the hairpins. Let me run that. I'm not so sure this will loop. So let me do it again. So there you see the green glow, and you also see the trapping on the hairpins. So now we have a way to look at the appearance of a messenger RNA in living cells. The second feature of this kind of work is that some of these spots are brighter than others, and that's because these spots are never, or not never, quite often there's more than one message there. So 2 have been made, or 3 or 4, so the spot is proportionally brighter, and that allows us to count the number of messenger RNAs that are there at any given time. And then we can do statistics, and we can begin to model the data. Here's another neat one. So this one is very similar to this, except if we look at the gene here, we also code, and our hairpin's down here. So this will be green then, when it's made. And we also put a red protein here called RFP for red fluorescent protein. So we can see both proteins being made, that is the translation of the message, and we can watch the message itself being made. And that's in this movie. This movie's a little more complicated than that, but notice, to keep an eye on these guys here. They're making a lot of the red fluorescent protein, and you're also seeing the message that's coding for that red fluorescent protein. Now, I always tell my students, if this doesn't excite you, then you should work in some other field. Because here you're actually seeing the central dogma of molecular biology going on before your eyes. You're not seeing DNA replication, but we could do that too if we wanted to. So what are we going to do with this now? Well, we're going to ask questions about when are they made, how stable are they, how do they get partitioned into cells as the cells divide, and so on and so forth. So is it clear what we're seeing here? This one's actually an IQ test, because you notice it's got mixed red and green. It's a little more complicated. So I'm not going to tell you what this experiment really wanted to show. Just want you to keep an eye on the red protein that's being made here. And it's being made here, but it's also being diluted by some green stuff as well. Okay. So here's a field taken from some time ago, and you can see the spots. And here's the spot intensity. And here's the number of particles that we find. So it's just a histogram of some data. And this is the number of GFP molecules per particle. And this was calibrated by doing in vitro binding to hairpins of different lengths. So we actually can now see, on average, how many protein molecules are bound to the message. And it's never 96, obviously. I don't think any of them ever managed to do that. It looks like it might be that way there, but. Okay, so now here's some data, real data taken from a paper some time ago now. But still, it's classic work. Take my word for that. All right, so now we're going to actually look at the mRNA, and we're looking at the protein, and I showed you how we did that. Here's one result here. These are the induction kinetics if you just look at this figure. So we induce the gene here, and in this case, we add an inducer that's an analog of lactose. So these cells are not making the message at all. They're completely quiet. You see no green spots. You see no red protein. And then you add the inducer at various levels and follow it with time. And you notice the first thing, it turns out that the central dogma is correct. The message is made first and the protein is made second. I always kidded Ito when he did this and said that, it'd be really exciting if it were the other way around when we took a close look. But no, this is what you do expect. It's not obvious, though, when you quantify this, what that relationship is, and that hasn't been followed up. So here's a typical picture again. You see some cells have green spots, but not much red. There's some red there, some red there. This one's making a lot of protein, and there's a lot of message, so it's been induced for quite a while. Now, the other interesting thing about this, which is the kind of qualitative part that one perhaps could stop at, but I'm not going to do that. Here's one that's green all over, but there's no spot. There's no yellow spot. Here's another one. So if you look at this a while, you can already see that your naive idea of how messenger RNA is made, which is you add the inducer, the polymerases all come to the promoter, and they all get started. There's something wrong with that right away. I mean, maybe the experiment's no good, but the experiment's okay. So basically, the simple model that there's an expectation with some fixed rate constant with time that you will bind the polymerase to the promoter and get going doesn't look as though it can be correct here for a couple of reasons. One of them, obviously, these guys didn't get going. And you could say, well, maybe they're not healthy, maybe they're dead, but you can follow them on the movie I just showed you, and you can show they divide normally. So there's no problem there. The other is that some of these cells have more message than others. You can argue, I guess, about how many and how big the difference is. But the fact is that some have more than others. Those that have a lot tend to be making quite a lot of protein. That's the red stuff, right? All right. Now, it turns out you can count the number of messenger RNAs in one of these particles. And the reason for that is that when you actually count enough of these and look at them carefully, you find peaks of fluorescence. So here's the estimated number of transcripts here, transcripts being the word for the messenger RNAs. 1, 2, 3, 4, 5. There's a distribution in count, but the important part is that it actually shows peaks. And it'll turn out that for the argument I'm going to make, if this is 1 or 2 or 3, doesn't really matter. It's important that it be integer, and that in fact you can tell this one from this one. Now we know from reconstruction experiments in vitro, that's sort of the biochemist in me, that in fact this probably is a single message, because we can take pure messenger RNA, we can add pure MS2 GFP, and now we get spots in a test tube, and we can look at the number of photons that come off under the microscope. So this is kind of an absolute quantification, and it actually seems to work even for quite large numbers. So that's just another way of taking this data and saying that there's, say, 10 samples or so that had just 1, some that have 2, 3, 4, and it's diminishing returns. And this is simply quantification to show that there's good agreement between what we claim from the microscope in the live cells and what we find by quantitative PCR and by another measurement done by another investigator. Okay, so far so good. Is it clear how we're going to do this now before I move on? Take more questions? I know I'm not that clear. Yes?
The reason it's spread out, is that because single molecules are diffusing? The pale green could have been localized into a few spots?
Like this?
Yeah.
So, yeah, okay, so this is where experimental stuff is so interesting to me. If you overproduce this from the plasma that I drew over there, it'll form little clumps because proteins tend to aggregate. So you've got to be very careful to induce the level just so that it's more or less everywhere in the cell. In fact, we repeated some of these experiments about a year ago in my lab. We had a lot of trouble backing off on the GFP level so it didn't aggregate. But it's pretty uniform. Yeah. Anything else? Yeah. Yeah. Yeah.
What I understood from this here was that it's the same transcript. This messenger RNA has these hairpins, and that's where the green stuff binds.
Yeah.
And it also codes for a protein, and it's to the protein that the red stuff binds. Is that right? The same?
No, the red protein is just made and it diffuses around freely. It is no relationship to this stuff here. They're made from the same messenger molecule, though.
They're made from the same messenger molecule.
Yeah.
Okay, yes, so that's right. The same messenger molecule which, as a messenger molecule, sends a green spot,
Yeah.
bright spot.
Yeah, yeah,
The same messenger molecule gives rise to a protein,
yeah,
which gives a red,
Yeah.
right?
yeah.
So now, where you actually see the green messenger RNA,
Yes.
why don't you also see the red there?
So that's a good question. So in lecture 2, I'll be talking exactly about that. So here's the simple reason is, the cell's only 2 microns long, and the diffusion coefficient of this protein it's about 25,000, its mass. And so the diffusion coefficient tells you from just the diffusion equation, that it should be everywhere within 10 seconds, if it's freely diffusing. And even if it's subdiffusing, which I'll show you it probably is, it doesn't change that by very much. So it just goes off shoom like that in this small space. There's no way to keep it there once it's made.
The red protein.
The red protein. It's not going to bind to anything. It's just a marker, really.
Yes, but because you had those green bright spots
Yep.
in those cells,
Yeah,
eventually that protein should have been produced.
and it is.
And but we don't see any red at all. I mean, there are some cells which just have green
Yeah.
and no red at all.
Yeah, I'll get to that.
Okay.
Now, one reason for that is we added the inducer here to turn the whole system on at 0 time. And so the question is, one question that we ask, and we will discuss it, is how long does it take before you should see anything? The old model, before we did these experiments, was that all of the promoters would be occupied. They would then all make messenger RNA in some kind of Newtonian way. They'd all be making it at the same time in the same amount. And so both the spots and the red should rise uniformly throughout the population. But it's not. It's a highly stochastic process. And I'll give you the answer, which I'll get to in a moment. It actually, the synthesis averaged over the population looks like a Poisson distribution. But when you look at individuals, you discover that no longer fits. In fact, the model for this, which we'll get to, is there's a exponentially distributed wait time for something to happen. But then once it happens over and over again 4 or 5 times. So that once you've done something magic to this promoter site here in the DNA, it's opened up for some length of time, let's say. And then there's an on and an off. And now, during that length of time, polymerase can get started over and over and over again quickly, but it'll close again. So there's 2 distributions going on here at once, and that's actually what you're seeing here. So it's kind of you might deduce that just from looking at this without measuring anything. I don't think you could, because you could interpret this many ways. I don't know if that's a satisfactory answer, but basically, once the red protein's made, it's like light. It's everywhere in the cell, just a few seconds, and it's diffused away because there's nothing to hold it there. Now, I'll contradict myself and say that with the lac repressor, when we make it at one location, it actually forms a gradient away from that location. But that's because we could look at the 1-second time scale. And you just can't do that with these kinds of experiments. Here you have E. coli growing, and you pipette in your inducer. By the time it's mixed and everything, there's already time goes by. But in any case, even if you could shorten it, just a thought experiment to try and answer your question another way, if you could shorten your observation time after you added this inducer, and then it got started, if you could look in the first few milliseconds, you would see the red protein where the green dot was, because that's where it's made. But in fact, by the time you've waited a few seconds even, it's diffused uniformly throughout the cell. There's nothing to trap it. Whereas, of course, with this green protein, it gets trapped here with that message. Does that get closer to a...
Okay, maybe we'll discuss it.
Yeah, okay, there's another question. I don't want to go too quickly. And you may think this is too much experiment for a theoretical group, but I'll do some theory next. Yeah.
Yeah, so the bit of the RNA that codes for the red fluorescent protein. So
Yeah.
how does the ribosome know when to stop the synthesis of the red fluorescent protein? Because it has these additional bits of RNA.
Sure, well,
beyond that.
it
So-
just comes, the ribosome just comes chugging along, and then there's a code word here that says stop, and
Oh, okay.
and it
so
keeps right
all
on
of
going.
The,
yeah,
the
it goes
headers
right
are
on,
beyond that. Okay.
right out through the
Okay.
end. Yeah. Okay, so I've I think we now have an opportunity to understand this. Okay, so here's some induction kinetics and population averages. And basically, this is data. There's a fair amount of scatter in it. And it's average. So this is the average N molecules, time after induction. And there we just look at several hundred individual E. coli as a function of time under the microscope, just like the last picture that I showed you. Experimental data, and this is a simple model. So we're approaching a steady state here. So we add a little bit of the inducer, and the cells get going. They start to make the message. And then they can only make a certain amount. There's finite machinery, right? And the induction level is only so high. So they reach a steady state, and it takes them about an hour under these conditions. This is room temperature. And then after that, this would be the simple model for this. They can't do any more. Can't make any more. They're not going to. Excuse me. I hope I don't lose my voice completely. Here's a more detailed model of this phenomena, and here's a simulation. The way we model it is to say that there's a constant rate of production, K1. So it's a very simple equation that you can teach freshmen, I know, having done it. We talk about the rate of synthesis of the average number N here as a function of time. Here's a rate. This is K1 is simply you make so many molecules per second, and then it gets degraded. So if this were a turnover question, if the protein were turning over, to get back to another question from the audience, this would be the degradation step. But in fact, it's immortalized here, and that allows us to do something quite interesting with the data. This is essentially, this term happens only when the cells divide. So that if a cell has 10 of these bright spots in it, and it forms daughters, then the daughters can get 5 and 10 and nothing, 9 and 1, so on and so forth, and we've measured that. There's a binomially distributed distribution between daughter cells. So they just get partitioned out. And that essentially is all that ever happens to this message. It doesn't get degraded, doesn't turn over. So we don't have to worry about that. So we can set this up, and it turns out that that's this particular fit here for this data. When you solve this equation, you end up with this function just equaling e to the minus k2t. It's the only variable we have to fit. This is, we measure this one. We know from bulk, we measure this one. You can get it directly from this kind of figure. Okay? So that's what happens in a bulk rate, and we actually come up with transcription factor rates and so on. This is a fit here, transcription rate, K1, K2 is a dilution rate, just the division time, and this is log 2 over 50 minutes, which is how rapidly they divide under our conditions. Yeah?
So transcription rate should be proportional to the MRNA concentration. So what is the rate?
Yeah. No, not the protein, just the RNA. We're only looking at the appearance of the counting spots, basically. And the intent. So we see a spot, and we say there's 2 messages there. There's using that other calibration curve. Yeah. Another question back there?
So isn't the mRNA level rate the function of the inducer?
A function of what?
Inducer itself.
Yes, it is.
Then why do we take a constant K1 there?
Well, because this is done under one induction
Only
regime.
one. Yeah.
Like, I forget what it was. We worked all the way from 1 micromolar up to 10 millimolars of inducer. So you can add very little, or you can add a lot. You see, so there's another question. I put it in your reading, actually. So Ito's group has followed up on this, and they've looked at more than one gene in E. coli. This is all done with the lactose promoter and operator. He's looked at 20 or 30 others now, and shows that, in fact, this, and no surprise to any of you, that this the stochastic look to the data is a function of how many message molecules you make. So if you make a ton of them, then you either can't see it anymore or it's not there anymore. And I think people disagree about that, actually. I don't, but okay. So this is the this is fine. This is consistent with the Poisson process. And I want to show you some data. So to my way of thinking, this, thinking of it mainly as a biologist, here's one of the extraordinary things about the data set. So if you just look at a plot of the number of message molecules per cell this way, and normalized frequency here, and then time after induction here, you find, not surprisingly, that initially there's 0, right? Very few. This is a big data set. And then with time, we march through this way. But after 2 hours, look at this tremendous heterogeneity in copy number per cell. And this we're going to interpret now as a simple idea that you first get on with the polymerase. But once you're on, or once the state of the DNA is changed, then the probability of making another message and another one goes up. And then that probability decays with time too. So we have two distributions going on. One, the probability distribution of a simple Poisson process to find things, and then what happens after you've found them, okay? So this actually turns out to be a simulation based on the model I'm going to describe for you. Okay, so this is the central idea, or the central piece of data that comes out of this. So here we've plotted the probability of 0 events. So at 0 time, before you add any inducer, none of the cells have any of these bright spots. None of them have message, right? And then, as a function of time, you ask how quickly do cells now appear with 1, 2, 3, 4, 5, n numbers of message. And here's a simple idea for the old model for this. It should fall off at this rate because we got the transcription factor rates from the bulk data, and that was the slide before, so we know the kinetics. There doesn't seem to be any ambiguity there. This is what it should look like, but in fact, this is what we discover when we look at individual cells. Okay, so there's this departure from a simple model. The Poisson, I don't know, I can never remember whether this is a Fano factor or not, but it's a variance over the mean. So for a Poisson process, variance over mean equals 1, as I'm sure you all know. The actual measured sigma squared over n for our data set is 4. And that led us to the conclusion then that once you've done something magic to the DNA so that transcription can get started, you can do it on average 4 more times to make 4 more message molecules. So in this really simple system, we have what turns out to be much more complex events going on at the molecular level. Now, what else? So this is just another way of plotting the same data. And if we go back, using this particular model, we can simulate the data very nicely, I think anyway, this data set here. All right, so that's how that story goes. And here, in fact, is the data that a biologist can understand. So I like to show this one if I'm not talking to anyone about kinetics. So let's just follow single cells now under the microscope and add inducer at this time here. And here's 1, 2, 3, 4 different observations. There's a quiet period. We don't see a spot, and then suddenly we see 1. There's a long time, and then there's 4. Here the cells divide. So this cell got some number. This cell, probably none, or we couldn't follow it is actually what this means. This particular cell then, nothing for a while, and then 2. So that's the data set you were looking at in the previous figure. Here's one almost 30 minutes after the inducer was added, nothing has happened. And then when it happens, there's a burst of probably 4 or 5, I can't. The resolution isn't very good here. There's a lag period, and then up it goes. Cell divides, and we start all over again. So from this, we can also get time on and time off for the apparatus itself, for the promoter, the RNA polymerase complex. And we get the distribution of burst sizes here. The expectation of almost any model would be that it's exponentially distributed, so that the chances of getting 8 or 9 or 10 is small. The most frequent ones are around here, 2, 4, and the mean is around 4. Okay, so that's the model based on the data. And I'm going over it to show you that the kinds of things you can discover by looking carefully in a new way at E. coli, basically, and then applying some simple analytical ideas to the data set. All right. Questions about that? Any answers about it? Conclusions? So this is now has been widely observed. I also gave you some reading in eukaryotic cells. Although I have to say that there's not much analytical work being done and no modeling. People observe it. It's called bursting in the literature, people who study this kind of stuff. And it's widely believed to happen for any number of genes in eukaryote as well as prokaryotes. But I think this is still one of the only quantitative studies to try and match the model to the data. So here's another one that's not quite as satisfying, but this comes from another Alberts et al. Volume, the big one. And it's the lambda repressor. And I'm going to talk a little bit about that later, so I'll just introduce it with this figure. How many of you actually know how this circuit works? I mean, anyone that studies gene regulatory circuits certainly must know. Better. Anyway. So basically, lambda has its incredible properties of using a repressor molecule. It's a virus, first of all. It integrates into the E. coli genome as a single copy, sometimes more. But it sits there and is passed on generation after generation as just 20-odd genes, something like, no, it's more like 120. As Jacob pointed out, there must be something that represses it so it doesn't get loose and replicate as a virus and kill the cell. It does that, but it does it at a frequency of about 1 in 10 to the 9th cells in the wild type. So it's got very tight control over whether it can multiply or whether it lies dormant, integrated into the DNA. At the heart of this is the lambda repressor called C1 here. This particular repressor has two qualities to it. One is it can sit and keep things turned on or off. I don't think I'll have time to go into this, but basically, in one state, it sort of prevents RNA polymerase from copying all the genes that will lead to cell lysis and death. And in the other state, it promotes replication. And in this particular case, there's another protein called CRO, that's this little guy here, that once you've managed to bump this off, CRO can then activate and transcribe, make transcripts this way, which launches the whole virus into a new life cycle. There's also DNA looping involved in this, and I think Rob Phillips is going to give a talk, and he'll surely talk about this. So I won't. But anyway, that's roughly the story. And it's a nice one to look at from our point of view, and so I'm going to show you some data to show bursting at this system as well. So this is the lambda PRM promoters. PRM just means it's a maintenance promoter. It keeps things quiet in the cell so the virus can't replicate. And here's some of the early data showing bursting as well for this particular system. So here's a system where the number of molecules, messenger molecules per cell is a function of time. And in this case, we're actually inducing a system that's quite complicated. But you can imagine there's bursts. They're not really all that happy. Ido's actually refined all of this data now, and he's got another nice example of it. Okay. So this is pretty much a summary of what I've been saying. The promoter activity, it's a Poisson process with a rate K1. At each transcription event, there's a burst of transcripts made. So you get on, and then you make something that has a mean of about 4 or 5 here. The result of activation and inactivation, this is something we don't know anything about. It's speculated that in higher cells it's probably modification of histones, but that's what a biologist would say anyway. Everything's going to be a modification of histones at the moment. E. coli has histone-like proteins, so it's not implausible, at least, but we don't know. And I think it's plausible because we can think of conformational changes. We can think of one of these other factors available or not available or concentration dependent or something like that. Okay, so that's the bursting molecule, the model, and that's the data. So I'd like to take any questions you have now or talk some more about it, because I want to turn to another subject that starts related to this but quickly diverges. Yeah, in the back. Is there water around, by the way? I'm going to dry right out any moment.
In the mRNA burst model, how do you account for the copies of the gene? I presume that this mRNA has been introduced into the cell through some plasmid. So are you tracking the plasmid copy number or something?
So that's a good question. I'm not so sure it matters, but the simple answer is it's a single copy number plasmid that behaves like the host genome, so that E. coli replicates, usually has one fork under slow growth conditions and has more than one under faster conditions. And this plasmid behaves the same way, but of course sometimes it will be making, just before doubling, it will be making 2 copies of everything and not just one. But I don't see how that influences things, because there we're only making the detector molecule. The message itself is coming from the genome, from the chromosome. I don't know if that answered your question, but anyway. Yeah.
So presumably the reason why you are seeing the huge spot over there of green is because there are multiple transcripts hanging off the locus, the gene itself. Right?
Yeah.
So how would this change? I'm sorry, I may be getting a little ahead of myself here, but how would this change in a eukaryotic cell, where you have, where presumably you wouldn't have multiple transcripts hanging off, and how would that affect the kinetics of the?
Yeah, to go back to the Oscar Miller picture, because as soon as it's made, it starts to get translated. And so these all, with this particular gene under these induction levels, it's not like that picture I showed you where there might have been 20 transcripts being translated. Here we're just doing a few. And I think the question is, I think from our model point of view, it wouldn't make any difference, but I think from a measurement point of view, I don't think we could see it, basically. Because we lose the stochastic part of it. I mean, it would still be stochastic, but you've accumulated, who knows, 100 messages or something like that. But I guess I don't really know the answer to that. That's what I think. Yeah. Yes, they do. So that was yeah, so I showed one slide, but that was for hundreds of cells. Now your question is, I think if you look at individual ones and sum them up, do they converge on the same answer? And to that is they do, yeah.
Like in longer time limits, all 3 curves converge to the same value.
Right, right. That's an important point, actually, that they do. Yeah.
In one of your talks, you were showing distribution of times 2 exponential.
Yes, yes.
So can you relate that to 2 different processes taking place?
Yes,
this
indeed, we can. Yeah.
busting,
That's an important part of it, actually.
Yes.
So we have the on, that is the, we can get both the on and the off rates from just following the lineage of cells that have been turned on and watching them divide. I should also say that I didn't include a slide for this, but you can show that when they do divide, the partitioning is just a coin flipping process, basically. There's no lineage. There's no, the grandmother cell doesn't get all of them and the granddaughters have to start again. It gets partitioned up with like a true coin flip or rolling of the dice. Yeah. Yeah.
Yeah, the process of a burst, coming on, and then a period of silence where there's no more transcript produced. Presumably that is governed by some transcription factor, activating the the gene and
Yeah.
inactivating it.
Yeah.
So what is the actual transcriptional regulation mechanism for this particular thing that you're tracking? What is, I mean, is there one transcription factor that's controlling the gene? Are there multiple ones?
We need the so-called sigma factor for this, and we also need another one, the CAP protein, the catabolite activator protein. But we take care of that either by changing the promoter so that we can dispense with one of them. But in these experiments, we just got the conditions right so that both those were available. So they're always there, both the sigma factor and the CAP protein or the CRIM protein. So there's really three proteins. There's the polymerase, and of course it's a alpha 2, beta 2, gamma something. It's a multiple of different subunits. And then there's the CAP factor and there's the sigma factor. So there's about 8 proteins form this complex. But it could be that the sigma factor, has an on and an off rate that could account for the bursting. I could imagine that, but I don't think we know that. I mean, someone must know more about this than I do in the audience, actually. But is that?
Yes. So
the simple answer is we don't know.
Okay. So some genes are constitutively expressed.
Yes.
And some genes are, under the control of, various sets of
Yeah.
transcription factors and
Yeah.
so on.
Yeah.
The particular one that you are studying, would it be called a sort of a constitutively expressed?
No, it's inducible. Yeah, it's inducible.
It's inducible.
Yeah. And now you're going to ask about the constitutive ones?
Yes.
The answer to that is they show bursting too. But I'm a little less secure on my assertion there. I think they do. I'm not so sure that I'm up to date on the literature. But I do know that when you make many messages, like the ribosomal RNAs, you don't see bursting, and I suspect that's because there's an averaging effect, and you just couldn't hope to see it. But I think that's good. Okay, I want to get started on another subject, and it's all I can say about this is that it's, I think from my point of view, so I was the department had a dinner for me about a month ago because I abandoned the department and moved to physics, so I have my spaces in physics, and I gave my microscopes away and things like that. And so Josh Shavitz, I don't know if any of you know him, but he's in the physics department, Princeton, young guy, and he measures bending of polymers, and he looks at E. coli and asks what's the bending modulus of the cell, does real biophysics. And he said that I sat somewhere between condensed matter theorists out here and then evolutionary biologists over here, because I used just enough mathematics to explain the data, no more, and I did just enough biology to create something to be worth explaining. So, and this is one of the results. So I'm going to have to take I hope this works. I'm sure it will. Let me get started on the second topic. I didn't know how long this would take, so whoop. There's a lot of material here. So I want to turn to another subject that's, I think, pretty exciting. And it's been perking along for some time now, but I was at Potsdam a month ago, where there was a whole congress on the question of percolation lattices as models for the cell and the transition from one kind of diffusion to another. So I'm just going to tell you what the problem is, and I'm going to tell you a little bit of what we've done about it, but it's a subject that's really ripe for development. And traditionally, this has been followed by people taking a little bit of dextran with fluorescein on it and injecting it into a cell and watching it diffuse. So you put a single particle in that's really bright, and then you ask, is it Brownian motion or is it something else? And most people have found it as subdiffusive. Some of these experiments have been done with actin polymerized in the test tube, where you do the same thing. You put in some little fluorescent particle and watch it move. So that means, of course, that when you start to model how transcription factors find their target on the DNA, You can't just plug in the Brownian equation for diffusion. You've got to think a little bit more about this. It's not that simple. So I'm just going to show you some pretty pictures. This is actually done by an old colleague of mine from a long time ago. And it's essentially a single metaphase chromosome. The DNA is about 10 centimeters long. And they put it down on a cover slip, and they just gently tweaked it with some trypsin or chymotrypsin to lyse the histones, the scaffolding protein. And you can see that it spreads out. And they did this to ask whether, in fact, a chromosome like one of ours is a single DNA molecule or not. So they did a lot of these, and then they looked carefully to find free ends, and they found none, basically. So they concluded not that it was a circle, but that if it had free ends, it didn't have very many of them. They concluded it was one molecule, which has turned out to be true. But I show it here because now you imagine yourself, even if you blow this up into a nucleus, and now imagine the problems you face when you're trying to find a little sequence that's only 20 base pairs long. In this nucleus, which is in a mammalian cell, is pretty big. It's about the size, the whole volume of one of those amoeba that I showed you, 10 to 20 microns across. And of course, it's not organized as a chromosome. When it's most active, it's opened up and it's filling the whole thing, so-called interphase. And then there's pictures that I'm sure some of you have seen by Goodsell. They're done to scale. They're done with the best kinds of data available. He's a watercolorist, and he has this beautiful book. This is the smallest cell known as a mycoplasm, and he's put in a realistic way all of the molecules that we know about for mycoplasma. Not very many. They can get by with about 600 genes, it turns out. Here's his E. coli, and here he's packed the chromatin in the middle, and he's put all of the proteins that are in the cell on the outside. Even that is something that's not very well understood. And then this is just a blow-up of one part of them. So these are actually transcription units here. These are ribosomal genes being transcribed, one of them, second one, third, fourth, fifth, and so on. But in this mess, this great big gooey mess in the cell. And in E. coli, the problem is perhaps simpler or maybe more complicated, but it is also packed. So this is a thin section, electron microscope picture with a cell wall, and then packed in here, I don't know, hundreds of different proteins, and some of them there on a nanomolar concentration, and some of them there in a millimolar concentration. This is a recent simulation that I found in PLOS Computational Biology. It's a realistic drawing of something like 50 different proteins, of which you can find them all if you look at this long enough. But it's very crowded in there. And I like this particular idea, this particular way of looking at it. My students like it. I hope you'll like it. So this is the problem. So there's the transcription factor trying to find its way to a target. I mean, it's not going to be easy. I'm going to let the whole thing run. It's only about a minute. He does find the target. And it would be lovely if we could somehow do the real, measure it and actually see what kind of diffusion's going. There, no, you see, there's the problem. Anyway. So with this system that I showed you with the spots that are made, so we induce in the cell the MS2 GFP molecules, the fused protein, binds to the message, and now we can actually watch those spots move, okay? And here's some more movies, because this is the raw data. You can see this one is well formed, and now, I mean, anybody could do this. I mean, an undergraduate could easily now collect data and ask whether that fits a diffusion equation or not. And here's some more. You can also see, remember I told you earlier, this is probably a single message, and this is a group of 8 or so, something like that. This is burst, and this one hasn't. So there they go. So we measure all of this. And now we track these particles. And here's a particular track. And you can see that the motion's constrained and is punctuated by large jumps. So this particular particle in the x dimension did this, and the y it didn't move much, and so on and so forth. Okay, this is another way of plotting the data. And the surprise that came from this particular experiment was that here's the message made in the test tube. So we copy the message from the plasmid piece of DNA with RNA polymerase, and then we take the pure green protein that'll bind to the hairpins, and we just watch it diffuse under a cover slip in a microscope. And it fits the diffusion equation very nicely. So it's just the mean squared displacement versus the log of the time. But none of these other particles fit at all. They all show an exponent in the diffusion equation, not of 1, but of 0.7, which is a characteristic signal for subdiffusive behavior. There's always roll-off here, and there's good evidence now from other experiments done in other labs that this roll-off is a boundary condition. So you could even see it if you looked at those other movies. It comes to the end of the cell and bounces back again, basically. So we come up then with the idea that in this crowded cytoplasm, things can't diffuse as fast as they normally would. Oh, this was my contribution to this paper, actually. I'm always suspicious of data sets where people say it's this. And in biology, if you do accurate measurements like this, then you can actually scramble your data set. You can randomize it and ask if the answer is any different. So this is just essentially the difference between the trajectories that this data, and then this is the randomized trajectories here. And this is actually a frequency plot. So here's the actual equation that fits best for the data. And it's a long-tailed distribution of waiting times. And this is actually, I don't know how many of you know Klafter. You probably do. He's at the Weizmann Institute. He did a lot of early work on, Levy distributions in biology, for example. And he's the one that gave me the Charlie Chaplin movie. So, okay, so what hinders the motion? Well, if you saw those other pictures, it's easy to imagine. In our case, we did several additional experiments because with E. coli, you can depolymerize the actin network. You can get rid of these histone-like proteins, and none of them actually influence the result. Yeah?
In the last set of experiments, how will you distinguish whether it's because of the confinement effect or it's really because of the subdiffusive behavior?
I'm sorry, could you just do this first part again?
So when you say that the exponent is different from 1,
Yeah.
subdiffusive behavior,
Yeah.
how will you distinguish whether it's because of the confinement effect or it's really subdiffusive behavior? I mean, boundary conditions.
Yeah, so I don't, well.
Am I not clear?
You think it's just bouncing off the walls all the time to give that, the subdiffusive coefficient? I mean, that's your, is that a hypothesis?
No, if a particle which is diffusing inside some confinement, like
Yeah,
in some confined region,
yeah.
so in the long time, the behavior of mean square displacement could be something like
Oh, okay, so.
how will you distinguish?
I don't think we can. So here's how I look at it. I see this as like a percolation lattice, basically. This is what I think the cytoplasm is like for a diffusing molecule. But in fact, I think you're saying, but maybe you're not, that the actual diffusion coefficient could change as well. For example, this is known to be true for people that set up real percolation lattices in engineering labs. If you put temperature differential across the lattice, you also begin to get, you begin to change the exponent. So I think that the activity of the protein in the thermodynamic sense, if it's changing, that can do it as well. And so it's not the model that it's just the packing of the spheres around it is certainly not the only way it could happen. No, I didn't answer your question. Let's try again.
So what I'm not clear is if there is, you are looking at the mean square displacement as a function of time.
Yeah.
So if it is linear, you will say it's a diffusive. If it is the power e to the power alpha is less than 1, you will say it's a subdiffusive.
Yeah, yeah.
So it can be because of really Levy flight kind of distribution.
Yeah.
Or it can be because of the boundary conditions.
Yes. So the question is it boundary conditions, right?
How will you distinguish whether
Well.
it's because of this or that.
So I think here we're seeing the boundary conditions where alpha is now starting to change even more. And here I think it's not, I mean, just the microscopy tends to show that this is not going to be a very good argument, but the microscopy shows that it's jumping around. It doesn't seem to be hitting anything, but I have to tell you that when you're looking at E. coli, it's only a micron thick, and it's awfully hard to know whether it's close to the cell wall up here or down here. So I think probably that might be a weakness in the argument. You're the first to point it out, though. Yeah. I think the other things, the actual mechanism, I don't know. There's another question back there. Yeah.
Yeah, it's more of a continuation, really, or a comment related to the same thing. So
Yeah.
I guess the confinement really is, I mean, the roll-off is indicative of the confinement, because even if you look at the free curve
Yeah.
in vitro,
Yeah.
then even at very small times it is diffusive. So
Yeah.
in the cell environment, before it starts feeling the wall,
Yeah.
if it were really just like a simple fluid inside, it would show diffusion at that time.
Mm-hmm.
So it's very clearly the subdiffusion is non-trivial.
Mm-hmm.
So
I think so. I mean, I
So
agree with you.
that's-
Yeah.
yeah, and
Yeah.
the second sort of question that I
Yeah.
had is, what's the spatial resolution that you have for the displacements?
Well, you probably can guess, if you didn't already know, that for a particle that size, that's that bright, you can get down to an angstrom if you wanted to. You just look at the mean, grab frames for as long as you can, and then you can get very high accuracy. But we didn't do that. These frames are gathered at basically video framing rate, which is 30 hertz in my lab anyway. I think it's 30 hertz. So the time resolution is not. Does that fit with what the graph
No,
actually
the spatial
says?
resolution, not the temporal resolution.
Yeah, no,
The
I was just
smallest-
thinking that if you get good time resolution, you also get good spatial resolution because you define the mean of the particle with greater and greater accuracy. But
Yeah,
the
but
spatial is going to be pretty good. I mean, if we go back and look at this, you see that particle is about, to be very conservative, it's about a 50th the length of the cell. So if you just sit on it and get it, if you grab, so every second we're grabbing 30 frames here. So I think
Yeah.
that it must be good. I can't tell you how good though.
yeah, I suppose you could just extrapolate the MSD curve to 0 time and
get
look at
it
that
that way.
value. So that's
Yeah.
probably
Yeah.
right. Thanks.
Well, that's good questions. Thank you. Anyone else? Yes.
I just wanted to comment on the point which he brought about. Is it also possible, I don't know if this is very trivial, is it also possible for you to have your control experiment in a confinement?
Yes.
So that you can just get the diffusion coefficient in
Absolutely.
that case? Okay.
Yeah, we could easily do that. We could put the synthetic stuff in a
Right, yeah.
small oil droplet. I mean,
Yeah.
in a small water droplet, and oil is one
Yeah.
good way to do it.
Yeah.
Haven't done it. It's a nice experiment, though. Be easy to do. You should do it. I'll send you the strains. And you do it just by putting a little, mix it and then squirt it with a syringe into oil, and you get little droplets the size of E. coli, and some of them will have something in them. Oh, wait, this is important. And it turned out that this is also a small theoretical literature built up around this. So in this is a PRL paper, and we did this sort of hand-waving thing and then made a reference to Digen's, who was always doing kind of hand-waving arguments, nonetheless very effective ones. And we pointed out that subdiffusion makes the problem of finding a target on the DNA better, not worse. Because although it takes longer, you don't escape into the 3 dimensions as easily. And in 3 dimensions, it's almost impossible to come back to the same spot. That's sort of the hand-waving argument. And now, this particular Weiss has done quite a bit of this now and actually shown fairly convincingly by simulations that subdiffusion helps the target finding problem in the cell as well. So that's what I wanted to conclude with that. Okay. I think I'll spend 10 more minutes on this because this leads me to another question. I don't know how many of you are, how familiar are you all with the literature on how repressor molecules find their target, or how any molecule finds a target, I guess, of any kind in the cell? Is this there's 3 famous papers from von Hippel's lab. Berg's theorist, Winter was the graduate student who did the work. And they basically showed that when you look at the binding of lac repressor to DNA, that when you embed the target in longer and longer pieces of DNA, the problem doesn't scale with the length of the DNA. You would expect that when you have 100 times as much DNA, then it would be impossible to find that target, buried in this needle in the haystack. So their way out of it was to think that perhaps the repressor binds to the DNA and then diffuses in one dimension along the DNA until it bumps into it. So basically they're reducing the dimensional problem from 3D, the naive way of looking at it, to 1. And the fully blown theory has several things going on. One is hopping across adjacent strands, and one is, no, it's not shown here. Another one is, if you can imagine, this is a long way away here. This is a very big loop in the DNA. But if you just had a smaller loop, you can jump from one loop to the other. This is their model. It's had a very, very big influence on anyone who thinks about transcription. Here's another lovely paper from Warren and Ten Volde, in which they asked if how you organize your genes in bacteria matters at all. And so they used an evolutionary argument. They said that, well, if it matters, that is, if the genes that you're going to repress or induce or influence, like C1, which is right next to the thing it does in lambda, and the lac repressor, I didn't tell you this, but it's only 23 base pairs away from the promoter that it sticks to. 23, no, it's a little bit more than that, but it's very close. And so the question is, does the cell care, does evolution care about the distances between the genes that are regulated and the repressor that's going to do the regulation? And so essentially they just went through the literature and they just asked these are known interactions around the E. coli chromosome. And there's these blue spots are essentially the highly significant ones in which the regulatory molecule is right next to the thing it regulates. And this is a randomized data set in which I think there's only 1 or 2 things that pop up as being next to each other. So they took this Berg and von Hippel idea a little further and said that, in fact, this difficulty of searching, however you do the searching, is a force in evolution to organize operons, as they're called, like the lac operon, next to its repressor. Well, I think that this paper from MRNA really covers everything pretty thoroughly. This is just a question of watching the transcription slide, watching it hop before it gets off and back on again, and jump between strands. I recommend that paper actually for anyone. And here's a more recent one from Ralph Metzler in which they take into account everything, the whole DNA, RNA complex, the messenger RNA, the ribosome, everything you could imagine. And then here's one from Johann Elf's lab in which they begin to talk about crowding and the effect of crowding. And this paper asks, what happens if these nonspecific proteins, like a histone protein, or in bacteria they're called HU proteins, they're bound and they give the chromatin some structure. What influence does this have on target finding? So here we have a repressor coming along. It can get on. It can diffuse in 1D or in 3D. And then it runs into these every once in a while, and that prevents search. So it's become a crowded field. Now, what have I got here? Okay, I'm going to stop here because this gets to some recent work published in my lab with Tom Coleman, in which we actually took the lac repressor and we put it at 4 locations around the E. coli genome. So we had it near the origin of replication, we had it near the terminus, and we had it in the left half and had it in the right half. And then we took 14 different locations to put the target, so you can see how you would do this too, right? You put the 14 target locations one at a time for these 4 different locations, so it's 14 times 4. And then you ask, does it matter to the cell? And you measure that just by looking at control of the lac operon. So does distance matter? And you can do that in a living cell. Just measure the amount of enzyme that's made as a function of separation between the repressor and its target. That's a really simple experiment. Turned out to be a whole year's work just to learn how to put genes where you want them around the genome, because these are big pieces of DNA, and we solved that problem, or Tom did. And then the next part of the question is, if you label the repressor in these same strains and follow it in the cell, can you actually watch it diffuse from where it's made to where it binds? We haven't done that yet, but we have a trick. So I'm going to talk about that tomorrow. I'm going to stop for today. So that's where it's going. But crowding is in here too. Ah, okay. Good. I'm not going to tell you why, but it's in there. It converged. It converged on the same question, actually. Because basically, you won't have to come now. I'm going to tell you the lesson, what we've discovered. We discovered there is a gradient of the repressor from where it's made to the other parts of the cell. So it's sort of back to your question about the red protein. It actually, you can see, it's not the best evidence I've ever published, but I think it's really solid. It's not a huge effect, but in terms of the dynamics of the cell, it's a huge effect. It's just not quantitatively huge. I shouldn't be talking about the weaknesses in the data now. Wait till you see it. And that's what I'll do tomorrow. And we model it, of course, so we have models for it. And we've reduced all of this complexity of Berg and other people to a simple 1D model. So I recommend you come back and see it. Any other questions about this? Yeah.
About 4.
Yeah, so that's a really good question. If you read the paper, you'll see that there's some deviation from the average data set in the newborn cells, the tiniest ones. And we speculate about why that might be, but it's not a big one. But there is it may not even be true, but you can see an inflection in the data. But one of my main points tomorrow is going to be that there's a profound difference between crowding in the newborn cells and then their parents. And this really has to influence transcription, but we haven't been able to show that yet.
Yeah, so one more question. So the other thing, probably I just was looking too much at the pictures. The bright spots were normally at the edges of the cell, at least in the beginning. Does that have anything on how the polymerase find DNA?
Yeah. So basically, I have 2 answers to that. One is that's true. They're rarely in the middle of the chromatin. And then the other answer is Tom Coleman has now thought of a way to see them when they're first made. And there you'd expect they'd be right in the DNA, buried in it, right? And he does. But the funny thing is that most of the time, they don't come out very easily either. So they tend to get trapped. So these are examples of ones that were moving too. So the data set's kind of biased, really is. I don't know if you know what Occam's razor is, right? You've all heard of Occam's razor? No? Well, Occam was a British monk who said that hypotheses and explanations should be sufficient to explain the data, but no more than that, right? You don't want to have this crazy rig with arms and legs when you only need one finger. Well, Sidney Brenner came up, he's a famous molecular biologist, with Occam's rug. And that's where you sweep data that you can't understand. You sweep it under Occam's rug. So I think in all of these, all this thing about it. So this is another thing. I could get going on this because most of you are theorists, right? You got to be very leery about experimentalists because they've got notebooks full of things they can't explain. And some of it is shocking. It's the only word for it. They're pretty stories, but there's this big mound under Occam's rug in the lab. Yeah, there's another question back there. Yeah.
Yeah. So is the diffusion that you observe anisotropic in X and Y, or is it isotropic?
It's isotropic in X and Y, yeah. That was a power spectrum I showed actually well, maybe I didn't show it in both the X and the Y dimension. The Z one is the tough one with fluorescence because in the Z axis there's this huge cone of light that comes out, and the X and the Y it's usually about a third the uncertainty that there is in the Z. Oh, sure, absolutely. And in fact, I built a microscope, but I gave my microscope away, so I'll never use it. But so here's the way you do it, and it's actually, I think, beautiful. And Tom Coleman has taken the lens with them. So you put a cylindrical lens in away from the focal plane, and it causes an astigmatism at the focal plane, so that spherical objects appear spherical when they're right in the plane, but they appear as an ellipse when they're out, and the ellipse grows more elliptical as you get further away. But the cool thing is that it's this way when you're above the focal plane, say, and it's this way when you're below. So now you can do very fast data gathering because you don't have to move or scan. You could just use the microscope we've been using and slip the lens in. And I know it works, but we've not got any data with it yet. Okay? Well, good. Tea. Tea and honey. Okay, well, thank you all. I'll be back.